All right, today is going to be kind of an interesting way for me to do this. I am recording the lecture through Zoom, and I'm actually in a different room. So if it sounds kind of echoey, that's why I have to have three devices open, and I've never done genetics, obviously through distance learning. So this will be a little bit of a challenge. So what you're going to want to have next to you, obviously, are your notes. So your guided notes you're going to be filling out, but then you also want those genetic problems that I posted that I said needed to be printed out. So they definitely need to be printed out because working out genetics problems are pretty difficult to do with typing. And I didn't make them editable just because the formatting is so weird. So please print it out. If you don't have a printer, you will need to work them out on a piece of paper then, but I am going to reference the problems. And so you're going to want to have them open somewhere, but I really prefer everyone print those out. All right. I'm using a different device here, so please have patience with me for this lecture. I am going to be using the Zoom whiteboard as well as PowerPoint and trying to follow along with your fill in the blank notes with you while also working out problems. So hopefully this will go okay. But just a little history. This guy right here on the side, his name is Gregor Mendel. He was an Australian monk, and he studied pea plants. And he studied the inheritance. There should be an I right there. That's misspelled. There should be an I. So he bred pea plants to study inheritance. And the reason why he wanted to study pea plants is because he could control the mating. He didn't have to worry about a pea plant getting up, walking over to another pea plant and mating with it, so he could control it. There is also many varieties available, and I'm going to show you a chart in a minute that shows you all the different characteristics that a pea plant could have. And you're going to know so much about pea plants after this, it'll blow your mind. Pea plants actually have flowers, and they have white flowers, purple flowers. The peas can be wrinkled, they can be smooth, they can be yellow, they can be green. So many varieties. So he had a lot of traits that he could study. And then they have a short generation time. So from the time he breeds them to the time where he can actually see the offspring was a really short time. All right. So you want to write that down. If you're not done writing, go ahead and pause because I'm going to flip to the next slide. We're going to go over some vocabulary here. All right, so the P generation are your true breeding parents. So they're the ones that he starts with. What we mean by true breeding is this purple flower only has genes for purple. When this purple flower got two sets, right? So two genes for each trait, one from what's considered the maternal, one that's considered paternal, that one only has purple, okay? The white flowers are pure white, only came from all white ancestor flowers, okay? So the P generation, the parental generation, are considered true breeding, purebred. They don't carry any other trait but the one that they're showing. So when you cross this purebred purple with the purebred white, you get all purple plants or all purple flowers. That's called the F1 generation. The F1 generation is the offspring when you cross the two P generations, all right? Then when you take two F1 flowers, two purple flowers from F1 and breed them against each other, you end up with this result in the F2 generation. The F2 generation is then used to figure out what is the mode of inheritance, what trait is dominant, what trait is recessive, etc. And so what I'm trying to do here is I'd like to annotate. So I want to kind of work this out for you guys in case you don't really have a good background in genetics. So when you follow this down, can you look at it and can you determine what's the dominant trait? Is it purple or is it white? Well, if you see right here, if you're crossing this purple and this white and you get all purple, you can determine purple is the dominant trait. So right here, you're getting, I would say big P for the purple times little p for the white. We have determined, because if you cross a purple with a white, you get all purple offspring, that purple is dominant. And so you're going to assign the big letter to purple. So then what are the letters for the F1 generation? Big P, little p. All right. And then if you did a Punnett square, and I'm trying to get this to look thinner. If you did a Punnett square where you cross those two, so remember Punnett squares from middle school, you would put a big P here. Oh, this isn't working that good, but it's all I have at this point. Little p here, and then what you would fill in is big P, big P, big P, little p, big P, little p, little p, little p, and it fits, right? So if purple is the dominant trait, anytime you see a big P, it's going to show the dominant trait. And so if you look right here, I'm going to change to yellow. If you look right here, that has a big P, that's going to be purple. That has a big P, that's going to be purple. That has a big P, that's going to be purple. And then this one right here is little p, it's going to be white. So what you can conclude is 75% of the F2 generation is going to be purple, 25% of the F2 generation is going to be white. All right, hopefully that was clear. This is usually how I would do it up on the smartboard at school. All right, so I am going to move on to the next slide, if I can. Okay, an allele. So what you're seeing here are two chromosomes. All right, these are two chromosomes. Blue means it's the paternal, came from dad, it's the paternal one. Red means it came from mom, it's the maternal one. And what you see is something called an allele. Alleles are alternate versions of a gene. And so when we're talking about the pea plants, right now we're talking about flower color. And so the purple flowers is an allele. It's a type, it's a version of a gene for flower color. White is an allele, it's a version of a gene for flower color. So alleles are alternate versions of a gene found on a chromosome. One allele comes from mom, one allele comes from dad, and they can be different from each other, kind of like these flowers. Some are purple and some are white. Okay, here are all the different characteristics of pea plants that Mendel studied. And this is what he found from doing those crosses. What he found to be dominant and what he found to be recessive. So when a trait is dominant, it means if even one allele of the dominant trait is there, the dominant trait will be expressed. So kind of like the Punnett square that we did. If a big P is there, it doesn't matter if there's two big P's or there's one big P in that outcome for the Punnett square. If a big P is there, the flower color is going to be all purple. And so the same thing with flower position. Axial is on the side, terminal is on the top. It looks like axial is dominant. Yellow is actually dominant to green when it comes to seed color. Round is dominant to wrinkled. Inflated pod, constricted pod, green pod, yellow pod, and then tall is dominant to dwarf. And he found the exact same pattern of inheritance for all of these traits. And so he came up with these principles. So alternate versions of a gene called alleles cause variation in inherited characteristics among offspring. So you're going to fill in the word allele. For each character, every organism inherits one allele from each parent. So one is maternal, one is paternal, you have two. That's why anytime you give the letters for a trait, it's either big P, big P, big P, little p, little p, little p. There's two of them. One came from each parent. If two alleles are different, the dominant allele will be fully expressed. The recessive allele will have no noticeable effect on the offspring's appearance unless you have both recessive alleles present. And then the law of segregation states the two alleles for each character separate during gamete formation. So that's why we're able to do a Punnett square, and we're able to give probabilities based on that Punnett square, because it's completely random. And I'll show you in a minute, it's completely random how chromosomes get sorted into gametes. And so this is what's kind of, it's showing you. So this is showing you that Punnett square I did for you a minute ago. So if you didn't understand it, this is another time for you to look at it. Phenotype is the physical trait that's being shown. Genotype are the letters. And I think I'm going over those words here in a minute. I hope so, at least. And then I'm going to move on. All right. So big P is dominant. Little p is recessive. Oh, here it is, phenotype and genotype, but this picture actually isn't on your notes from what I can see. So, so genotype and phenotype, you need to know the difference between those. Dominant, recessive, you need to know the difference between those. Homozygous and heterozygous. Homozygous is where both letters are the same type of letter. So if both are capital or both are lowercase, that's homozygous. If you have one capital with one lowercase, that's heterozygous. So homo means the same, big P, or little p. Hetero means different, big P, little p, okay? And so on your homework, what I'd like for you to do right now is look at your genetics practice problems. Number 1, for each genotype below, indicate whether it is heterozygous or homozygous. And you can put he or you can put ho. So we'll do the first 4 together, and I'm moving down, A, B, C, D. So big A would be homozygous. Big B, little b would be heterozygous. Big B, little c, heterozygous. Big D, little d, heterozygous. Big E, little e, heterozygous. Little f, homozygous. Hopefully you're following along the homework with me. If not, you need to pause and go back and get the homework. Big G, homozygous. Big H, homozygous. And then I'm going to let you finish filling the rest of those out. All right. Number 2, it says for each of the genotypes, that means for each of the letter combinations, determine what the phenotype would be. So let's do the purple flower are dominant to white. Purple flowers are dominant to white. Big P would be purple. Big P, little p would be purple. Little p would be white, right? And then I'm going to let you fill in the rest of those. Okay. Number 3 says for each phenotype below, list the genotypes. Remember to use the letter of the dominant trait. So if straight hair is dominant to curly, it's wanting you to list the different genotypes that could give you straight hair. You can pick any letter you want. I prefer to pick letters where the uppercase looks a lot different than the lowercase. So for hair, I would do the letter H. So for straight hair, if straight hair is dominant to curly, straight hair could either be big H, or it could be heterozygous, big H, little h. Curly hair is going to be little h. All right, and I'm going to let you do the tail spikes. I would use the letter T and not the letter S, because in uppercase S looks too similar to a lowercase s and you would get yourself confused. So definitely get into the practice of using letters where the uppercase and lowercase are a lot different from each other. Okay, and let me see where I want to go from here. Like I said, this is kind of a difficult, Okay, I don't want to move on to test cross just yet. So let's look at number 4. So I'm going to kind of help you set up number 4. And then after that, you should be able to do all the rest of the problem. So let's do number 4. So what I'm going to do right now is I'm going to go to my whiteboard and we're going to look at number 4. So the first one is big R, little r, little r, little r. All right, so this looks like round seams are dominant to wrinkled. And these are the two parents that we're going to cross, and the question wants us to tell it, or the question wants us to answer what percentage of the offspring will be round. And then I look back and I'm like, wait, is round dominant or recessive? Okay, round is dominant. All right, so then I make my square. All right, and then I put my letters for this parent first, big R, little r. Then I put my letters for the second parent, little r, little r. And then I cross them. So where these two letters right here meet up, I'm gonna write big R, little r. Where those two meet up, big R, little r. Where these two meet up, little r, and then where these two meet up, little r, little r. So what percent is round? 50%. Okay. So I would just pause if you don't have that done yet, because I'm about to erase. All right, erase, erase, erase. All right, hopefully when I play this back, the sound works and everything, because I think this is actually working out pretty well for my first time. Okay, I'm gonna do the second one. The second one wants you to cross two heterozygote parents. Let me get my pen ready. Big R, little r, big R, little r. So those are two heterozygous parents. I'm going to draw my box, and then I'm going to put the letters on top. Big R here, little r here, big R here, little r here. And then I'm going to fill in my boxes. Big R, big R, big R, little r, big R, little r, little r, little r. Okay? What percentage are going to be round? So what percentage of these offspring are going to express the dominant trait? Well, how many out of 4 have a big R? 3 out of 4. So 75% are going to be round. Okay, pause if you didn't get that, because I'm about to erase. Erase, erase, erase. Okay, this is actually kind of fun. What if I end up liking distance teaching better than I like regular teaching? Just kidding. Never. I would never want to just teach without students in front of me. Okay, but it's really cool to have these kind of tools, and I'm discovering all these new things that I can use to try to make it less painful for everybody. So this is the next problem right here, big R, which is homozygous dominant, times big R, little r, which is heterozygous. I draw my Punnett square. I put my letters on top. Big R, big R, big R, little r. And then I fill in my boxes. Hopefully I'm not going too fast. Just kind of pause if you're like, wait, where'd she get that from? Just kind of pause. Okay. And then this question asks, what percentage of the offspring will be wrinkled? All right, round is dominant, wrinkled is recessive. Are any of these offspring going to show the recessive trait? The answer is no. In order to show the recessive trait, they would have to be little r, and there are no little r's, so 0%. All right. So at this point, I would want you to go through all of these problems. If you had, I'm still continuing on with the lecture, it's not over, but for homework, or it's all at home, but for outside work that's not guided by the teacher, you're gonna do all those problems, and then you're gonna ask me to get on Zoom with you, and I'll use this whiteboard if you need any help. I'm not sure if I have to erase this before I go back to my slideshow, but just in case. Just have a little clean workspace. All right, I am going to go back to my slideshow here. All right, so next on your notes, you have lots of pictures. There's just lots of pictures on here. And we are not going through this whole handout today. We're doing half of genetics. We're just doing Mendelian genetics today. And then we are going to do non-Mendelian genetics on the next day. So for some reason, I don't think you have this on your notes, but you already wrote this down, that phenotype is the physical trait, genotype is the genetic makeup or the letters, and then this is the example for that, okay? A Punnett square is a device for predicting offspring from a cross. And we just did a bunch of we just did several Punnett squares. Okay, we did 3, but I did another one. So we did 4 Punnett squares so far, and then you're gonna practice all the other ones on your own and ask me for help on Zoom if you need to. So that's why I would make sure I tried this and not be 11 p.m. at night when Dr. Holbrook's asleep. All right, a test cross is used to determine if a dominant trait is homozygous or heterozygous by crossing with a recessive. Okay, so here's where this kind of comes into play. I want to annotate here. All right, so let's say you have a purple flower. You have a purple flower. If you just look at the purple flower and you know it's complete dominance means if the dominant allele is there, it's going to show the dominant trait. So right here, if you are looking at a purple flower, you don't know if that purple flower is big P, or if it's big P, little p. You just don't know looking at it, and you don't know what the ancestors of the flower are. So what you would do to determine whether it was big P or big P, little p is you would cross it by something that you know. And what you know is if the flower is white, it has to be little p. So you'd cross it. If you got this outcome right here, that means that the original purple flower was big P. So if you got all purple offspring, that means the original purple flower was big P. If you got some white flowers as offspring, that means that the original purple flower had to be heterozygous for the trait. So that's a test cross. So a test cross is used to determine if something showing the dominant trait is either homozygous dominant or it's heterozygous, and then you do that by, you perform a test cross by crossing it with a homozygous recessive for that trait. All right, so hopefully that made sense. Oop, yep, I actually do have to erase. Well, now we know. So then if your other teachers are trying to do this, I feel so lucky that like I got to practice so much this summer with you guys and with my anatomy class. I feel like I'm just like so ahead of other people. But I can help other people if they need help. Like we had that breakout room party, Zoom party the other day with a bunch of teachers and showed them how to use it. All right, let me get off that toolbar. Okay, so we know about test crosses now. Law of independent assortment. So each pair of alleles is going to segregate separately, independently from each other. That means that no traits are linked together. So let's say the gene for hair color and the gene for eye color, they are not inherited together, supposedly. So at this point in time, when Mendel was figuring out genetics, he believed no traits are inherited together. So no 2 traits are ever connected together. I know that that's not true because green eyes and red hair are considered to actually be linked together. And they won't, like the green eyes and the red hair, the genes are so closely linked together that they usually won't separate from each other. And that's probably kind of a hard concept to understand. And what you're seeing in front of you right now is something called a dihybrid cross. A dihybrid cross is where you have 2 traits and you're going to do a Punnett square for it. And I'm going to show you in a minute how to do that. It looks really difficult. It's going to take some practice. So this is what your second set of worksheets is for, is a dihybrid cross. And so I'm going to show you that here in just a minute. But all I need you to know, law of independent assortment, no 2 traits are linked together and they will randomly sort into gametes during meiosis independently of each other. And so this is kind of showing you what are the 2 options. So homologous chromosomes can line up like this, or it's just as likely they would line up like that. And you end up with different outcomes, and each one of these outcomes are just as likely probability-wise as the other. That's what law of independent assortment means. Okay, so far we have looked at a monohybrid cross where we're studying only one characteristic, like flower color, and you use a 4-box Punnett square for that. All right. A dihybrid cross is a Punnett square where you're studying 2 characteristics at one time, like flower color and seed shape. And I'm about to show you how to do that. It is a 16-box Punnett square, supposedly, but I have shortcuts. I'm going to show you the shortcuts. All right. Here's an example of a dihybrid cross. This is where I usually start losing students. So if you need to pause and then come back for this, this is going to be a little intense. So you might want to take a little break. All right, dihybrid cross. I have 2 traits, trait A and trait B. I have 2 parents, parent 1 and parent 2. Parent 1 is heterozygous for both traits. Parent 2 is heterozygous for both traits. I see 2 traits are being used here to do a Punnett square. So what I'm going to do is I'm going to make 16 boxes. This is going to be difficult to fill in because I'm using my finger. So I'm probably going to have to use text boxes to fill it in. 16 boxes. Okay. So what do I put at the top? What I put at the top are what are the possible combinations of alleles that could be sorted into that parent's gametes. And so each gamete, remember, is haploid. It's only going to get one version of the gene. It's only going to get one allele. And so I have 2 traits. So what I'm going to look for is what are the different combinations of A and B that can go in, that can be sorted into the gametes. One option would be big A, big B, right? Another option would be big A, little b. Little a, big B, little a, little b. Do you see how those are all different combinations of A and B that can be sorted into the gametes of this parent? Then I do parent 2. Well, parent 2 is the exact same genotype. So the gametes options are going to be the exact same as the other parent. So I'm going to do it like that. Okay? All right. Then what we do is the exact same thing as before, but we keep the letters together. So if I am filling in this box right here, okay, this box right here, I'm going to put both of those A's together and both of those B's together. And this is where it's getting kind of difficult to use my finger to fill in such a small box. That's why I need a stylus. I just ordered one off of Amazon. Sorry. All right, then I'm going to fill in this one. Big A, big B, little b. Okay, do you see where we're getting those letters? You take the combination of letters that are on the top and the combination of letters that are to the left, and you combine them into the box. So this one would be big A, little a, big B, big B, big A, little a, big B, big B. And then I just keep going. Big A, big B, little b, big A, little b, little b, big A, big B, little b. There are students that actually love Punnett squares. I was one of them. It's hard to, it's definitely not the majority, but some students really do like doing this. It's usually the students that like math. You're just doing a process, trying to find an answer. All right, little a, big B, big B. Hopefully I'm not making any mistakes here. It's easy to make mistakes on these 16 box Punnett squares. Big B, little b, big A, big B, little b, and please excuse my handwriting. Little b, little a, big B, little b, and then finally, little a, little b, little b. Aw. Okay, and then what we do at the end, you're gonna love this because I'm about to show you a shortcut. You should never, ever have to do this again. Ever, 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 because there's a rule. Let me show you. All right, I'm trying to, hold on, I'm trying to find my drawing pen, and I wanna do it in yellow. Okay, so the question is, how many of these 16 boxes are dominant for both traits? How many are dominant for both traits. So I would pause right now, and if I were you, I would circle them. How many are dominant for both traits? This one's dominant for both traits. Remember, it only has to have one capital letter. That one has at least one big A and one big B. This one has at least one big A and one big B. This one has at least one big A and one big B. This one does. Does this one? Nope, there's no big B's. Does this one? Yes, there's at least one big A and one big B. Does this one? Nope, there's no big A. This one, this one. That one have both dominant? No, because it's not dominant for A. This one's not dominant for A. That one's good. What about this one? It's not dominant for B, so I can't choose that one. This one is not dominant for A, and this one is not dominant for either. So how many did I circle? 1, 2, 3, 4, 5, 6, 7, 8, 9. So 9 out of 16 are dominant for both traits. Right? How many are dominant for A but recessive for B? Dominant for A, recessive for B, dominant for A, recessive for B, dominant for A, recessive for B, and that's it. 3 out of 16 are dominant for A, recessive for B. How many are recessive for A but dominant for B? 1, 2, 3. And then how many are recessive for both? 1. Okay. Pause the video if you don't have those circles done. Here is the rule. Erase, 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 erase. Erase everything. Oh gosh, this is gonna take a second. Erase, 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 erase. Okay. Sorry, I know this one's taking a long time, but this is important. It's something I have to work out with you. Okay, so the rule is, if, and I want to write this down, if both parents are heterozygous for both traits, here's what's called the 9-3-3-1 rule. If both parents are heterozygous for both traits, 9 out of 16 are going to be dominant for both. So these are the offspring. 3 out of 16 are going to be dominant for the first trait and recessive for the second trait. 3 out of 16 are going to be recessive for the second. I should have done a text box for this. Recessive for the first trait, dominant for the second trait. And then 1 out of 16 is going to be recessive for both. Hopefully you got that down. 9 out of 16 are dominant for both. 3 out of 16 are dominant for one, recessive for the other. 3 out of 16 are recessive for the first one, dominant for the second one. 1 out of 16 is recessive for both. That's called the 9-3-3-1 rule. All right, if you don't have that down, go ahead and pause because I am going to move on, and then I'll work a dihybrid from your homework. So I think we're going to talk about probabilities here. Remember, I have to get everything erased. Actually, I'm going to just move on to one of your homework problems. Okay, so if you go to the dihybrid worksheet, let's work out the first problem for that one. Then I can finish up the notes here real quick. If I can find the dihybrid, I'm trying to find it on my computer right now. So find the dihybrid worksheet. Cell bio, okay. Punnett square practice, that's not it, dihybrid practice, here it is. All right, so it has a bunny rabbit on it, super cute. Aw, how sweet. Black nose, pink nose, long ears, floppy ears, how fun. All right, we have a male rabbit. Let's stop share. I'm gonna go to my whiteboard. All right, we have a male rabbit. Does everyone see where I'm at? It says genetic crosses that involve 2 traits, floppy-eared bunnies. In rabbits, black hair is dominant to brown. Long, straight ears are dominant to floppy ears. That's unfortunate because floppy ears are really cute. Just because something's dominant actually doesn't mean it's more common. There are some things where recessive trait is more common. These letters represent the genotypes and the phenotypes of the rabbits. Cool. First problem, a male rabbit with the genotype, big B, little e. So what does that mean? What does he look like? It looks like he has a black nose and floppy ears. I'm sure he's super cute. He is mating with little b, big E, little e. What does she look like? Aw, pink nose, long ears. Okay, so that's our cross. Okay, so we know since we're dealing with two traits, we're gonna need a 16 box Punnett square. Actually, let me make it a little bit smaller because I'm gonna have to fill it in on the top. We're gonna need a 16 box Punnett square. Okay, so you draw your 16 boxes. Now we need to figure out, kind of like we did before, what are the possible combinations of B's and E's for the dad rabbit? What are the possible combinations for him? Are there any little b's? Nope. Are there any big E's? Nope. The only possible combination for him is big E, little e. So I'm just going to go ahead and put that on top. The only possible combinations for him. All right. What about for the mom rabbit? She doesn't have any big B's. She only has a little b, but she does have a big E and a little e. So what we're going to do is we're going to fill hers in like this. That's a possibility. This is a possibility. And then if I use that second B, that's a possibility. That's a possibility. Okay. Let's say I start filling it in, and I'm going to use a text box for it. Let's say I start filling this in. Big B, little e, B, big E, little e. Cool. What am I going to put right here? I'm going to put the same thing. And what am I going to put right it all out? Why don't I just simplify it by, if I try to push erase, it'll erase all of it. Why don't I just simplify it by crossing this out, and this out. So instead of making it a 16 box, I just make it 4 boxes, 2, 3, 4, okay? You could simplify it further, but I don't want to confuse anyone even more. I'm just saying if you have gametes repeating themselves, you don't even need to do those boxes. All right, here I'm going to do big B, little e, little e. The text box here. I'm going to do big B, big E, little e. All right, and a text box here. I'm going to do, come on, move text box. Okay, whatever. Big B, little e. There it goes. Sort of. I'm gonna move that right there. Okay, and that's really all there is. And so what's the question asking? How many out of the 16? Well, we didn't really do 16 because we went ahead and eliminated those. All right, how many out of the 16 have black nose, long ears? So dominant for both. How many of these are dominant for both traits? I'm so glad they actually did the boxes for you. Dominant for both traits would be, draw, that one, that one. 2 out of 4, or had you filled in the whole thing, it would have been 8 out of 16. All right, so you can reduce it. 2 out of 4, 1/2, 8 out of 16, it's all the same thing. Black nose, floppy ears. Do we have any black nose, floppy ears? Here, black nose, floppy ears right there. Same thing. If you had filled in all 16 boxes, you would have found 8 out of 16. Or if you did it like this, you would have found 2 out of 4. Or if you even reduced it further, you would have found 1 out of 2. Pink nose, long ears, 0, right? Yeah, there's no pink nose, long ears because there's no little b anywhere. And then pink nose, floppy ears. Well, there's no pink noses anywhere, so that's 0 also. Okay, so what you'll need to do is try to do those problems. And you will have a quiz where I'll actually give you one of these problems. So make sure if you are having trouble understanding it, that you ask during the Zoom call for me to work something out for you or whatever, because I'm going to devote the Zoom call to working out these problems for you. No, I don't want it from this slide. Darn it. Resume. There we go. Okay. So then we can just have some probabilities. I don't think, I'm going to be honest here, I don't think I want to, I feel like I really gave you a lot of information, a lot of stuff to work out. So what I'd like for us to do, some of you will read, oh yeah, laws of probabilities, that's pretty intuitive. What I'd like for you to do is just cross out the part of your notes that is talking about laws of probability. So I'm looking at your notes right here. Probabilities, so that entire section, just go ahead and erase it. All that is, and if you really want to fill it in, you can. I'm not going to hold you responsibility for that. So rule of multiplication means two events are occurring independently of each other. So if you're throwing dice, let's say you threw a dice 2 times, what's the probability that you get 6s both times? You'll multiply 1 out of 6 times 1 out of 6. What's the probability of having 5 boys in a row? You do 1/2 times 1/2 times 1/2 times 1/2 times 1/2. That's a 1 out of 32 chance. These probabilities can be used if you're doing 2 different Punnett squares. So if you have 2 parents and they each are, you're looking at 2 traits, you would do a Punnett square for hair color, and then you could do a Punnett square for eye color. And if you want to know what's the probability that their child is going to have brown hair and blue eyes, you could do problems with that. I'm more concerned if you can just do 1 Punnett square, 4 boxes, and then if you can do a hot die hybrid with 16 boxes. I'm not going to throw probabilities at you at this point. So you can just go ahead and skip over that. Just skip over it. Those of you that want it though, the next one is rule of addition. If you have an or, so what's the probability that the offspring will have brown hair or blue eyes? You would add, okay? And I would ignore that. We're not going to look at that. All right. And then tomorrow, what we're going to do is non-Mendelian genetics. I think I've given you enough for today. So please have all of those problems worked out, and you're going to take pictures of your work and you're going to upload them to Teams, and then let me know if you need help, and we will discuss them in the Zoom tomorrow before you take your quiz. All right. I am going to stop recording now.